Hello dear participant, we are in SWAM course. Course name is Introductory Animal Physiology, Digestive Physiology of Monogastric Animals. Title is Digestion of Nutrients. We are in week 5, lecture number 2. Hi, I am Dr. Manish Kumar, Assistant Professor in the Department of Veterinary Physiology and Biochemistry, Faculty of Veterinary and Animal Sciences, Institute of Agricultural Sciences, Banaras Hindu University. So we are going to talk about the adaptations that increases the small intestine surface area, the structure of the villus, what are the different structures are present in the villus, and the muscles contractions within the gastrointestinal tract, and eruption contraction of stomach as well as esophagus, and digestion and absorption of the different nutrients in the stomach and intestine. Let's start with one by one. First, we'll be talking about the intestine. Intestine is hollow sac-like organ and it is thrown into fold and then villi and microvilli. This fold, villi and microvilli are mainly there for the increase of the surface area. So if you talk one by one, that is fold, that fold increases the surface area by 3 times of the intestine. If that fold is not there, surface area will be decreased by 3 times. And then villi increases the surface area that is thrown over the fold, that is situated over the folds of the intestine. It increases the surface area by another 10 times. And the third one is microvilli, which is hair-like projection known as microvilli, arises from the luminal surface of the epithelial cells, increasing the surface area another 20-fold in a formation called the breast border. Villi are longest in the jejunum and progressively decreases in the length through the ileum. Altogether, that is fold, villi and microvilli increases the surface area of the intestinal tract by 600 times. If you are coming to the structure of the villi, how the villi will look like. The absorption across the digestive tract wall involves the transepithelial transport, similar to the movement of the material across the kidney tubules. The major component of villus are epithelial cells that covers the surface of the villus, which forms tight junction and limit the passage of the luminal content between the cells. These epithelial cells have within their luminal breast border carriers for absorption of specific nutrients and electrolytes from the lumen, as well as the breast border digestive enzymes that complete carbohydrate and protein digestion. Second component of the villi is a capillary network, which is running to the each villi and it is supplying the blood, which is helping in the absorption of the nutrient. And third component is a terminal lymphatic vessels, that is called as lacteal. That is a terminal, it goes, and this is involved in the absorption of the fat. During the process of the absorption, digested substances enter the capillary network as well as in the central lacteals. Now we will be talking about the muscles contraction within the gastrointestinal tract. In the gastrointestinal tract, there are 2 types of muscles are there. One is circular muscles and another is longitudinal muscles. Circular muscles involve in the segmental contraction, which is helping in the mixing of the food particles, and these mixing and grinding actions facilitate contact between the digestive enzymes and the ingesta. It also constantly moves the lumen material against the mucosal surface cells that will absorb the nutrient. The peristalsis propels the bolus of ingesta down the gastrointestinal tract aborally. Aborally means away from the oral cavity. This requires coordinated contraction of the outer longitudinal muscles and inner circular muscles just behind the bolus and relaxation of these muscles layer just ahead of the bolus. The stomach musculature has an additional layer of the muscles layer inside the inner circular and outer longitudinal layer of the muscles, and it runs transversely in between these two muscles layer. This oblique muscles layer provides the another dimension for the stomach contraction that greatly increases mixing activities within the stomach. Contraction of the stomach begins at the mid-fundus as peristaltic wave that propels the ingesta towards the small intestine. Pyloric sphincter, controlling the movement of the material from the stomach to the small intestine, relaxes momently, means for a period of small time. A small amount of the most digested and liquid material, that is chyme, passes into the duodenum. The pyloric sphincter quickly contracts again before the peristaltic wave has been completed. This causes the more solid material within the chyme to hit the pylorus and be shunted back towards the fundic area of the stomach. This creates churning action that helps break down solid in the chyme, ensuring the good mixing with the stomach acid and proteolytic enzymes. Also causes the dietary fat to form the emulsion with the water in preparation for the digestion in the intestine. Stomach motility is increased by the hormone gastrin synthesized by the enteroendocrine cells in the pyloric region of the stomach in response to the distension of the stomach. When the food particles enters into the stomach, then this creates distension, and then gastrin, that is hormone, is being secreted. The vagus parasympathetic also respond to the distension by increasing stomach contractility. Vagal parasympathetic stimulation of the stomach contractility can also be initiated by higher center in the brain in response to sight, smell or taste of the food. Stomach motility is decreased when the chyme enters the duodenum. So, stomach motility is when it enters the duodenum, the stomach motility decreases. Vagal sensory efferent returns this information to the medulla, causing the reduction in the vagal parasympathetic efferent stimulation of the stomach contraction to hormone synthesized by enteroendocrine cells in the upper duodenum. These enteroendocrine cells are located in the upper duodenum and secretes the hormone, also act on the stomach muscles to slow the contractility. So, these hormones secreted from the upper duodenum is decreasing the stomach motility. These are cholecystokinin secreted in response to the purgence of the fat and amino acid in the duodenum, and secretin released in response to the reduction in the pH in the duodenum. These hormones are secreted into the blood and the portal circulation, eventually reaching the muscles to the stomach. These hormones also act on the pyloric sphincter and causes it to constrict more tightly to reduce the further entry of the chyme into the duodenum. So, once that this stomach content is coming to the duodenum and the fat, protein and acid content of the chyme leads to the secretion of the two hormone, that is a CCK and secretin from the enteroendocrine cells of the upper duodenum, causes the closure of the pyloric sphincter as well as it reduces the stomach motility, so that next batch of the chyme should not enter into the duodenum until the fat, protein are being digested. The contraction and relaxation of the pyloric sphincter determines the rate of emptying of the stomach. The pyloric sphincter relaxes in response to the vagal stimulation and contracts in response to the CCK and secretin. High fat and protein diets greatly increases the CCK secretions, slowing the emptying of the stomach, whereas high carbohydrate diet do not stimulate the CCK secretion at all. Now coming to the erection contraction of the stomach and esophagus, that erection contraction is important because gases are being swallowed through the food as well as the fermentation taking place in the stomach. So that gases are very important to be removed from the stomach, and that are being removed by the erection or burping contraction. Now we'll be talking the digestion of the nutrients in the stomach and intestine and their absorption, that is the digestion of the carbohydrate, digestion of the fat and digestion of the protein as well as their absorption. Let's start with the digestion of the non-structural carbohydrate. The plant starches and glycogen from ingestion of muscles and liver comprises large number of glucose molecules linked together by a bond at the alpha 1,4 or alpha 1,6 position. The most common plant sugar in the diet is sucrose, composed of a glucose molecule linked to a fructose molecule. Milk sugar or lactose is disaccharide of glucose and galactose linked by beta 1,4 position. On ingestion of a starch in the diet will begin to be broken down by the salivary alpha amylase. This process breaks some of the alpha 1,4 linkage, but the enzyme does not have time to break down very much of the starch molecule before the bolus enters the stomach, where the amylase will be destroyed by the low pH. So salivary amylase will be destroyed by the low pH. It will be acting only the pH is neutral pH is there. So if you see that non-structural carbohydrate digestion, mainly dietary carbohydrates is having the starch and glycogen. These starch and glycogens are polysaccharides. So once that animal eat the food and it is in the mouth and digestive tract lumen, so animal once they will eat it, they will chew the food and they will break down the food, they will lacerate the food. So this increases the surface area of the food, and in mouth salivary amylase will start working on this and they will start digesting, though it is not playing the main role in the digestion, but the digestion of the carbohydrate will start here itself. And then later on, that once it will enter into the lumen, so that pancreatic amylase will be acting on that and they will digest and they will convert these polysaccharides into disaccharides. So these polysaccharides will be converted into disaccharides with the help of salivary amylase and pancreatic amylase. This will convert, this starch and glycogen will be converted into the lactose, maltose and sucrose. So this is disaccharide. Now that these disaccharides will be moving towards the breast border of small intestine epithelial cells. Okay, so this will move to there, and there in the breast border of the small intestine epithelial cells, there will be the enzyme that is a lactase, maltase and sucrase. These three enzymes will be there and they will digest lactose into the galactose and glucose. It will be digested by lactase, and the maltose will be broken down into the glucose, two molecules of glucose by the maltase, and sucrose will be converted into glucose and fructose by the enzyme sucrase. So now these are the glucose, galactose and fructose are monosaccharides, and these monosaccharides are the absorbable unit. So they can be absorbed in the cytosol of the epithelial cells. So this is the absorbable unit of the carbohydrate. So coming to the now that food particles has been nutrients has been, that is carbohydrate has been converted into the absorbable unit. So now absorption will start. So dietary carbohydrates is presented in the small intestine for absorption mainly in the form of disaccharide, that is maltose, sucrose and lactose. I told you disaccharide is located in the breast border of the small intestine cells. This further reduces these disaccharides into the absorbable monosaccharide units that I told you, glucose, galactose and fructose. So glucose and galactose are being absorbed by the secondary active transport, in which symport carriers such as sodium, glucose co-transport on the luminal membrane transport both the monosaccharides and sodium from the lumen into the interior of the intestinal cells. The carrier which do not directly use the energy themselves depend on the sodium concentration gradient established by the energy consuming basolateral that sodium potassium pump. Glucose or galactose concentrated in the cell by these symport leaves the cell down its concentration gradient by facilitated diffusion via the glucose transport that is called as GULT2 in the basal border to enter the blood within the villus. A significant amount of the glucose crosses the epithelial barrier through leaky tight junction between the epithelial cells. Fructose is absorbed into the blood solely by the facilitated diffusion. It enters the epithelial cells from lumen via the GULT5 and exits the cells to enter the blood via GULT2. Now coming to the digestion and absorption of the fat. So we'll be talking about the digestion and absorption of fat. We have already told you how that bile is making this fat into the micelles. We will be talking about that. Dietary fat are generally in the form of triglycerides. Their digestion may begins in the mouth as a lingual gland produces the pharyngeal lipase that converts the triglyceride to fatty acid, monoglyceride and diglyceride. These enzymes are relatively stable in acid and is thought to play a role in digestion of milk fat by neonates. That may not be producing the full complement of pancreatic enzyme and bile from the liver. The amount of the fat is digested by the pharyngeal lipase has negligible effect in normal fat digestion. The first step in the fat digestion takes place in the stomach, where the dietary fats are subjected to the churning action of the stomach contraction. This causes the dietary fat to form emulsion with the water, a suspension of the fine droplets of the fat in water. The presence of the fat in the duodenum elicits the secretion of the CCK. I told you the presence of the fat and protein is the main stimulus for the production of the CCK from the enteroendocrine cells of the upper duodenum. So once this fat will enter into the duodenum, so this will elicit the production of the CCK, and now this CCK will go via blood to the pancreatic cells, and these pancreatic acinar cells will produce the enzymes for the digestion of the fat. And this CCK will also act on the gallbladder to contract, and bile salt will enter into the common bile and pancreatic duct to the upper duodenum, and this will involve in the digestion of the fat. So CCK, several critical enzymes can be found in pancreatic secretions, that is a pancreatic lipase, colipase, procolipase, phospholipases, cholesterol esterase. Bile contains the bile salt that play several roles in the fat digestion. The emulsified fat droplets entering the duodenum is too hydrophobic, too large for the pancreatic lipase to access the droplet and begins to break down the triglyceride. Bile salts are essentially detergent produced in the liver by combining cholesterol and amino acid. One end of the molecule composed of the cholesterol moiety is hydrophobic in nature, which allow it to form ionic bonds with the hydrophilic fatty acid, the hydrophobic fatty acid. The other end is a very hydrophilic is due to the amino acid component. Bile salt surrounds the emulsive fat droplets and break it into smaller fat droplets suspended in the water in lumen. This increases the surface area available for enzymatic digestion of the triglyceride to occur. Colipases must bind to the pancreatic lipase to allow it to be fully active, and this complex then begins to digest triglyceride to monoglyceride and 2 fatty acid at the surface of the intestine, small fat droplets created by the bile salt. Cholesterol esterase may free cholesterol form from the droplets, and phospholipases liberates the fatty acid and monoglycerides from the phospholipids. As the lipolytic action of these enzymes progresses, more bile salt surrounds the liberated fatty acid, monoglycerides, cholesterol, fat-soluble vitamin that were in the diet to form the small bile salt covered structure that is known as micelles. So, this is the structure of the micelles. You can see that it is covering the fatty acid and monoglyceride and cholesterol. So, this forms the micelles. So, coming to the absorption of the fat. Fat must be transformed from the watery chyme through the watery body fluid even though it is not water soluble. So, the fat, though it is not water soluble, but it has to travel through the watery body. Therefore, fat must undergo a series of transformation to circumvent this problem during its digestion and absorption. When the stomach contents are emptied into the duodenum, the ingested fat is aggregated into large oily triglyceride droplets that float in the chyme. The products of lipase digestion, monoglyceride and free fatty acids are also not very water soluble, so very little of these end products of the fat digestion can diffuse through the aqueous chyme to reach the absorptive lining. However, the biliary component facilitates the absorption of these fatty end products by forming the micelle. Once these micelles reaches the luminal membrane of the epithelial cells, the monoglyceride and free fatty acid passively diffuse from the micelles through the lipid component of the epithelial cell membrane to enter the interior of these cells. As these fat products leave the micelles and are absorbed across the epithelial cell membrane, the micelles can pick more monoglyceride and free fatty acid which have been produced from the digestion of the other triglyceride molecules in the fat emulsion. The bile salt continuously repeat their fat solubilizing functions down the length of the small intestine until all the fat are absorbed. Then the bile salt themselves are absorbed in the terminal ileum by a special active transport. This is an efficient process because relatively a small amount of bile salt can facilitate the digestion and absorption of large amount of the fat, with each bile salt performing its pairing functions especially before it is absorbed. Once the fat is within the interior of the epithelial cells, the monoglycerides and free fatty acids are resynthesized into triglycerides. These triglycerides then form into the small droplets and are coated with the layer of the lipoprotein, which renders the fat droplets water soluble. The largest of the lipoproteins, known as chylomicrons, are extruded by the exocytosis from the epithelial cells into the interstitial fluid within the villus. In mammals, the triglyceride-rich chylomicrons are secreted into the central lacteals and enters the lymphatic vessels, whereas in the birds, they are absorbed in the capillary of the villi. Fatty acid with the short or medium length carbon chains also enter the blood. Fat absorption is considered to be passive process. However, the overall sequence of events necessary for the fat absorption does require the energy. Now coming to the digestion of the protein. Protein in the diet tend to be very large molecules and often consist of hundreds of amino acids linked by peptide bonds. Casein, the major protein in the milk, has a molecular weight of 23,000 and is about 200 amino acids in length. These molecules needed to be broken down to at least dipeptide and tripeptide level before they cross the enterocytes. Protein digestion begins in the stomach. Acid environment alone can hydrolyze some of the peptide bonds. Chief cells of the gastric glands secretes the pepsinogen. An inactive proteolytic enzymes, gastric glands acid mixes with the pepsinogen and cleaves of the fragments of the pepsinogen to form the pepsin, the active enzymes. Pepsin cleaves the peptide bond next to the hydrophobic amino acid with the aromatic side chain. Renin is the another enzyme produced by the chief cells, cleaves between the phenylalanine and methionine residues on protein and especially important for digestion of casein by neonatal mammals. Both pepsin and renin functions optimally when the pH is between 2 to 3. The proteins that were hundreds of amino acids long when they entered the stomach enter the duodenum, the fragments that may be 25 to 100 amino acid long. As peptides reaches the small intestine, they activate the receptors of the enteroendocrine cells lining the duodenum crypts, stimulating the secretion of the cholecystokinin, CCK. The CCK enters the circulation and reaches the pancreatic acinar cells and myoepithelial cells surrounding the each acinus. This triggers the pancreatic enzyme secretions into the upper duodenum via the pancreatic ducts. The proteolytic enzymes secreted by the pancreas include the trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase A and B. CCK secreted by the crypt's enteroendocrine cells in response to the peptide entering the duodenum reaches the villous enterocytes as well. This causes the enterocytes to secrete the enteropeptidase. It is also called as enterokinase into the lumen of duodenum. Now this enterokinase or enteropeptidase seeks the trypsinogen that has entered the duodenum and cleave off the fragments to form the active proteolytic enzyme, that is the trypsin. Now this trypsin will convert more trypsinogen into the trypsin, and other enzymes also will be converted into the active form. Each of the different proteolytic enzymes, this is trypsin, chymotrypsin, elastase and carboxypeptidase A and B, cleaves the peptide bond between the specific amino acids so that when the luminal phase of the digestion is completed, the protein has been converted into the peptide that are generally just 1 to 12 amino acid long. These single amino acid and long peptides then move to the breast border. They are very soluble in water and cross the unstirred water layer and entering the glycocalyx, adhering the microvilli, forming the breast border of the villous enterocytes. Several intestinal peptides project from the breast border into the glycocalyx, but these enzymes are not released into the lumen of the intestine. These intestinal peptidase hydrolyzes the peptide bond and reducing the length of the peptides to not more than 3 amino acid in length. Now coming to the absorption of the protein. The protein presented into the small intestine for absorption is primarily in the form of amino acid and few polypeptide. These amino acids are absorbed across the intestinal cell by symport, similar to the glucose and galactose absorption. The amino acid symport are selective for the different amino acid. A small peptide gain entry by means of yet another sodium dependent carrier in a process known as tertiary active transport. In this case, the symport simultaneously transport both hydrogen and peptide from the lumen into the cell, driven by the hydrogen moving down its concentration gradient and the peptide moving against its concentration gradient. The hydrogen gradient is established by the antipode in the luminal membrane that is driven by sodium moving into the cell down its concentration gradient and hydrogen moving out of the cell against its concentration gradient. The sodium concentration gradient that drive the antipode in turn is established by the energy dependent sodium potassium pump at the basolateral membrane. Thus, glucose, galactose, amino acid and small peptides all get free rides on energy expended for sodium transport. The small peptides are broken down into their constituent amino acid by aminopeptidase in the brush border membrane by the intracellular peptidase like monosaccharides, amino acid enters the capillary network within the villus. In addition to the dietary protein, in the non-ruminant, sub-indigenous proteins are also being absorbed like digestive enzymes, mucosal turnover and plasma protein. These are also need to be absorbed similarly as the dietary proteins are being absorbed, otherwise deficiency of the proteins will take place in the animal. So this is the table where you can refer the different nutrients, how it is being digested, what are the enzymes involved, what is the source of enzyme, what is the site of action of enzymes, and what is the action of enzymes actually, and then absorbable units of the nutrients for the all three nutrients, that is carbohydrate, fat and protein. So in this class we have learned about the digestion of the major nutrients, that is carbohydrate, fat and protein, and their absorption, how they are being digested and being absorbed, what are the different enzymes are secreted and helping in the digestion of the nutrients which is being absorbed and it is being available for the animals. Thank you.